Exploring metagenomics, unveiling the genetic secrets of microbial communities, step by step. Introduction. Welcome to this training video on metagenomics, a fascinating field in bioinformatics that delves into the genetic makeup of entire microbial communities. In this tutorial, I will guide you through a step-by-step process to help you understand and analyze the complex world of metagenomics. Step 1. Sample collection and preparation. The first step in metagenomics is to collect samples from the environment or organism of interest. These samples can come from a variety of sources, such as soil, water, or the human gut. Once collected, the samples need to be processed and DNA extracted for further analysis. Step 2. DNA sequencing. After extracting the DNA from your samples, the next step is to sequence the genetic material. This can be done using various sequencing technologies, such as Illumina or nanopore sequencing. Sequencing the DNA will allow you to uncover the genetic diversity present in the microbial community. Step 3. Bioinformatic analysis. Once you have your sequencing data, it's time to dive into bioinformatic analysis. This step involves aligning the sequenced reads to reference databases, performing taxonomic classification, and identifying functional genes present in the community. Software tools like KIME and MGRAST can help with this analysis. Step 4. Metagenomic assembly. In some cases, it may be necessary to assemble the sequenced reads into longer contigs to better understand the genetic content of the microbial community. Metagenomic assembly can reveal insights into the structure and function of the microbial community, providing a more comprehensive view of its genetic potential. Step 5. Interpretation and visualization. The final step in metagenomics is interpreting the results of your analysis and visualizing the data in a meaningful way. This can involve creating heat maps, taxonomic trees, and functional gene networks to help you understand the relationships between different members of the microbial community. Recommendation: for those in the bioinformatics niche, mastering metagenomics can open up a world of exciting research opportunities. By understanding the genetic makeup of microbial communities, researchers can gain valuable insights into the role of the microbiome in human health and disease, leading to potential new treatments and interventions. Conclusion. In conclusion, metagenomics is a powerful tool that is revolutionizing our understanding of microbial communities. By following these step-by-step guidelines, you can unlock the genetic secrets hidden within these complex ecosystems. While metagenomics has its challenges, the rewards of uncovering new insights make it a rewarding and impactful field in bioinformatics.